I'm Sharif Mansi, professor of biochemistry at the University of Trento, and I am the coordinator of the Protomet program through the IETN network. And what we do is we explore protometabolic pathways that may have existed on our planet in the past and use that to help explain why extant biology is the way that it is. We have an exciting group of research scientists throughout Europe, both here at the University of Trento in Italy, as well as throughout the rest of the European Union, plus, I guess, the UK these days. So we have groups also in Germany, in Spain, in Switzerland, and 2 in France. We are 7 units, each providing different expertise to this project, including one company called Elvisis. And it's a really exciting network because we have the opportunity to help train future scientists in this emerging field of prebiotic chemistry and providing, I think, something really quite new to the European space.
We learn in school and also at university how the universe was formed. We know all about the Big Bang theory and how Earth came to be established. We also learned how we went from very simple organisms into the array of very complex organisms that we have nowadays. However, what we don't really know is how we went from a lifeless Earth to a world full of life. And that's what we're trying to answer. We are the chemistry PhD students of Protomet, and we're trying to bring light into new reaction pathways that could relate to an early version of metabolism.
And we are working in biochemistry field. We are studying how compartments are correlated to prebiotic chemistry.
As you know, compartments like cells and subcellular structures are very crucial for facilitating metabolism in contemporary life.
Thus, my work mostly consists of computational modeling of biological and chemical systems. This also includes the integration of experimental data potentially coming from my colleagues within this network. Therefore, my role is also to engage with all the other members of this project, trying to play some kind of connecting role between the different approaches represented within this network. All in all, my project aims at investigating what are the main criteria in order to characterize a biological chemical system as a metabolism, as well as trying to simulate such a simple protometabolic system.
My project was to apply microfluidics to develop complex microcompartments that could eventually further the artificial cell-like systems research field, and also to control the environmental parameters of biological systems, something that could potentially be used to investigate the environmental parameters of the origins of life.
One of the things that I really enjoy about this program is that there are few opportunities for students to get in, let's say, at the ground floor in this field at such an early stage of their careers. And so we are really, I think, one of the first to provide an actual degree program to be exposed to prebiotic chemistry, and I think this will situate these students well in this emerging field so that they can not just contribute intellectually, but also advance their postdocs, academic, as well as industrial careers later. So I'm particularly proud of this aspect of Protomet.